Dr. Valentin Bruttel, welcome to Weird Souls. Thank you for making time to discuss some very interesting and complex things together from your location, I believe, in Europe. And could you kindly introduce yourself for the audience?
Yeah, thank you very much for having me. I think it's very, very important that, especially in Europe, there's more investigative work done on COVID and that the audience learns more about this topic. In my opinion, the data that we have been fed with and major media has been pretty one-sided. But before we get into this, as you said, let's have a very brief introduction. So I'm a molecular um, biologist, basically, from what I do at work. And I apply that technology in the field of immunology mostly. I'm the CSO of a startup. I also work at the clinics, university clinics here in southern Germany, in Würzburg. But none of the views or things I say here are in any way related or representing my employees or my company. This is totally my personal, all my personal views. And I'd also like to say briefly that I have never worked in any gain of function virology projects. I don't know any virologists who do this kind of work personally. So I think it's pretty safe to say that I have no conflicts of interest. I've held two presentations on this topic at academic conferences, and it's just been such a big issue. And because we use exactly the same molecular techniques they use to produce viruses, to produce new therapeutics for autoimmune diseases, I think I know quite a lot about um, the molecular engineering and the construction of the viruses, not so much about how all the different viral strains are related. For that, you probably have to talk to a virologist.
So to begin this travel into where we are right now, so we're in the sort of mid part of 2024. Um, what does a scientist like yourself in Germany doing your own thing, being involved in your company? How do you get to look into the pandemic and what's going on early on? So maybe you came to it in 2029, in 2019, 2029. Hopefully not. Maybe you came to it very, very early, even before 2019. Maybe you came in when it affected you in Germany and you had lockdown, etc. Tell us that history.
Yeah, so I mean, directly at the beginning, it was just a few headlines in the news. I wasn't really following this as much as I've probably should. Um, and even before, there was this very brief gain of function research being discussed in the media here in 2014, 2013, when they did the gain of function on the much more lethal H5N1 bird flu viruses in the Netherlands, which is very close, obviously, and then in the US. But also back then, I kind of underestimated the importance of and danger that comes with this kind of science. So in the pandemic, I first started to become a bit suspicious when the first articles were printed that immediately said it's a conspiracy theory that this could have come from a lab, while it was really obvious they had a big lab working on exactly these viruses there. And then these articles basically didn't provide any good scientific arguments to support the claims. Yeah. But for a little bit, I was still saying, oh, well, these are five Western professors. They're not under the influence of any, communist party. They're not afraid for their lives. They say this is pretty sure it's from nature. Then this is probably true.
And so, are you referring to the Lancet Commission, or are you referring to the paper, or are you referring to the proximal origin? Which
Yeah.
of these exactly are you referring to that convince you that if those people are saying that, it must be true?
Yeah. So basically, there were like three key papers. I think the biggest one was Proximal Origins. It was the most cited one by far. Um, Nature Medicine is a highly um, reputable publication, very strict publication guidelines usually. Um, and I was especially shocked to read their internal communications later, where they basically say that they believe the opposite to be true of what they wrote. Um, and then the Lancet was a brief statement. And in that also we know that the first author afterwards in an email said that he can't exclude that this was a lab, or this would be stupid to exclude that. So to me, this was basically, science was kind of my guiding principle somehow for achieving the truth, and I was generally trusting fellow scientists, to be honest, and to really, of course they could make mistakes, but to print what they believe to be true. And this was quite revealing and shocking to see in two major journals that the opposite happened here in this case. Yeah, and then of course the science just didn't match their statements. This got me a bit suspicious, and I started looking a little bit deeper fairly early on into the engineering behind these lab-made viruses, how they are assembled, and if this could leave any traces. Um, yeah, in the genome. Of course, there's other pieces of evidence that point towards the lab. So there was this recent article by Alina Chan, which brought up quite a few points in the New York Times. It was about time, I think, that some of the scientists on the opposing side of the origin debate were finally allowed to express their opinions and show their data in a major media outlet. And of course, this was also kind of suspicious to have the outbreak location in Wuhan. But we could go through these in individual steps and discuss for all the individual points. I have 5 more, so to say, about the molecular engineering mostly. And yeah, and I think this can help people to really assess the evidence individually. I prefer usually not to tell you my opinion that much and focus on the facts so you can form your own opinions. But maybe you can, I hope your audience appreciates that. I think there was a lot of opinions being communicated and very little scientific data so far. So basically, I think there are 2 key origin scenarios. And one is that there was an accident during a project very similar or to one described in a leaked proposal. It's called the diffuse proposal. It was leaked more than a year after the outbreak. So people kept it secret for a very long time. And it describes quite a few of the very specific features that we find in SARS-CoV-2 that make this virus very different from what we usually expect from a naturally jumping zoonotic virus. And then, of course, there could be a zoonotic event. We don't know exactly which species would be the intermediate host, but we know that this would have come from a bat at some point. Yeah, all the closely related viruses were sampled in bats. But the first point basically is the location. And there is in the diffuse proposal a map that really nicely shows this, shows in which areas you have the highest density of bats, which are the hosts of these viruses, the so-called horseshoe bats. And those are mostly in the very southern part of China and some other southeastern countries like Laos, for example. And the Wuhan Institute of Virology was sampling bats exactly in these areas. They had an abandoned mine shaft in Yunnan province where they had sampled the RATG13 virus, which for a long time was the most closely related relative. And then later on, a French team went to Laos. They were not allowed to go into China anymore for sampling viruses. And they found a virus called Banal 2052, which is a tiny bit closer related to SARS-CoV-2. But they also in that map show that Wuhan is in an area that's completely white. So there are absolutely 0 viruses expected in Wuhan. And some scientists argue that you need a major city to start this outbreak, but there's also a lot of cities with more than 1 million inhabitants in these areas where the viruses are. And we know that there are people there that, for example, dig out bat guana for put it on the fields as a nutrient, as a fertilizer, and those can have antibodies against related viruses. So we know that sometimes jumps occur, but they don't occur in Wuhan. Wuhan used to be the negative control also for these antibody studies, because they said this is the city where we don't expect any outbreaks and where people are never in contact with these viruses. Um, so this would be very different from what we know from other outbreaks as a first point. First of all, that the city is very far from the natural reservoir of the virus. And then secondly, that the virus got there without leaving any traces. So for example, in SARS and MERS, SARS-1, the first SARS outbreak, you saw several jumps from civet cats, for example, into humans. And you could basically trace different jumps over time and how the virus mutated and how the virus found in the civet cats directly matched the virus in the patient. So you can really conclude that this is proven beyond reasonable doubt that this is the intermediate host. And we don't have any of that. We have just this virus appearing out of nowhere in the middle of a area, a city, where there is absolutely no viruses in nature. There's no traces, but we know that there's an institute that samples in exactly these regions. So if they take a sample and put it in a plastic container, of course, it would not leave any traces. There wouldn't be any other people infected on the way. And then also it was a hotspot of coronavirus research, especially with a focus on bats. So actually, I put this into PubMed at one point. Just the name of the 20 biggest cities in China, mainland China, plus bat and coronavirus, and you saw that in average, there was 5 times more coronavirus research papers published between 2006 and 2018 in Wuhan than what you would expect from a city of that size. So if you hadn't asked me where I'd expect a lab leak of a bad coronavirus based on that data, it's 5 times more likely to happen in Wuhan than in
Hmm.
any other Chinese
Hmm.
city. Yeah.
Hmm.
This is not super strong evidence, of course, yeah, but it's kind of suspicious and something we have not really seen before.
Hmm.
So yeah, and then the
Did
second
you,
point. Yeah,
before you
sorry.
move to the second point,
Mhm.
did you notice at the time, or was it known at the time, that the actual laboratory in question also had a very suspicious database that just basically was taken down very abruptly, and that also it seems from other type of forensic investigation that they had some sort of issue with their ventilation, and that there was a number of very drastic work done around the ventilation of the lab. And even if I read correctly, correct me if I'm wrong, but I read that there was some school suspension at a time and some disruption around. So, were you aware of that information back then?
Yeah, this was circulating in Twitter, I think. Then after a while, not so much in the news here. We had actually one German professor who collected a lot of this data. He monitored basically search results, for example, from Chinese Google. What's the name? I don't know. Their basically for symptoms and traffic around hospitals and cell phone data. And a lot of this looked very suspicious. He got attacked in German media for that because he's not a professor
Indeed.
of virology. Yeah.
Yeah.
But the data he collected made a lot of sense. With regards to this, yeah, I've heard about the databases. I think there was more than one that was taken
Yes.
offline for a long time.
Yes.
Of course, this is also very suspicious.
Hmm.
Um, we don't know exactly when this outbreak started because, of course, it's easy for a country like China to manipulate the samples and the data if they wanted to do that. We've seen this before in other lab accidents, like in the Sverdlovsk accident, where the central government, the Communist Party, controlled the entire narrative. Um, so it could be that the pandemic started already in September, maybe, when this database went offline. But I'm not really the expert on judging this Intel intelligence. Yeah, but of course, it leaves open questions, right? Why would they take it off? Why would the WHO Commission not be allowed to access it, even if they were hackers? What they claim to be the case, it wouldn't make sense to not allow like an official commission like the WHO Commission to access all their data and look through their files. And
Especially one commission which had at the helm of it the very man who'd been there multiple times, in Dr. Peter Daszak. So that's even more bizarre.
yeah, yeah, absolutely. I mean, for context, it's probably important to know a few names here, and maybe one of the absolute central figures is Dr. Peter Daszak. He's now, I think, facing criminal charges even in the US for being dishonest. And I'm not 100% sure about this, but
Eco Health Alliance, which is the thing that he was running, which was obviously working in very close um, collaboration with NIID and NIH and Dr. Fauci. So he has been heard in under oath, and I think they're prosecuting. They certainly debarred Eco Health Alliance from receiving further funding from NIID. But in terms of his own, because he is so proximal, so to speak, to a lot of very important people, including Fauci, Collins, Ralph Barrick, the sort of inception team for the gain of function, on the U.S.
yeah,
side, not, but he was also very close with Xi Jinping and the Bat Lady, and maybe so those names are names that people should be
yeah,
well familiarized
yeah,
with now. But that too is highly suspicious. The fact that the actual WHO and then the Lancet Commission has on its book people who are exactly involved with that, and that there are no disclosure of that right
yeah,
off the bat. So if it is a pandemic and it's accidental and we don't know what's going on and we're all terrified, why aren't these people coming up to the forefront and saying, you know what, we're doing those things, we're doing gain of function, and we're doing it. Let's not go into motives, but therefore we know it's plausible and we should look into that as well. No, instead you've got this blanket, like, oh, we're not telling you who we are. If not for Diffuse, if not for all this work of people, sleuth, on the internet, scientists all over the world who
fantastic.
decided to say, now, who is Peter Daszak? I think to this day, we probably wouldn't know who he is.
Yeah, it's an absolute scandal that they put him in front of this commission. He's the principal investigator on the diffuse application. Yeah, Ralph Barrick is from UNC in the United States. He is basically the bioengineer, I'd say, with regards to synthetic viruses. He founded this entire new technology, especially for these coronaviruses, which are fairly large, with 30,000 letters. He invented a lot of, adapted a lot of technologies invented for other DNA manipulation procedures to virology to generate these huge viruses. Then Sheng Li is the head of the group that collected all these viruses at the Wuhan Institute of Virology. Um, and there, and Peter Daszak basically was providing the money, organized by EcoHealth in New York City. They applied this at the DARPA, so U.S. military, which is also by, I don't think this is a intentional release or bioweapon. I think it makes sense that the Chinese basically sent their weapon plans as a research proposal to the US military a year before the outbreak. Yeah, but all of this is highly suspicious. They didn't release this research proposal, which was basically uh, the key answer to why we could have seen basically the second point, the furin cleavage site, which was absolutely new about this virus. Um, and if they had nothing to hide, they should have done that immediately. They should have said, look, we were also asking about very similar viruses, also partly planned to work on them in Wuhan. This is what we have. These are our computers, our lab books. Please go through everything. We have nothing to do with this. Yeah? Completely different story. And I think people would have, of course, maybe been a bit suspicious, but if you then put in people in place that are trustworthy, and I have not directly involved with this, I think people would have trusted such a commission. While what they did is putting Peter Daszak there is an absolute no-go. It's like asking the head of the VW to investigate cheat devices, for example. Right? Everybody immediately sees that
Yep.
this will not lead to anything objective.
Yep.
Yes. Uh, yeah, I think this went really bad. And then we later also found out that Peter Daszak was behind that Lancet paper, where they also condemned this or condemned scientists who say this could have come from lab as conspiracy theories without saying that he basically collected these interviews. We know emails where he talked to his collaborators on this, Lin Wang and Ralph Barrick, saying that they should rather not sign it because that would look too suspicious. And it's just the entire thing has nothing to do with good, honest, transparent science, right? This is an absolute no-go. We know that some of the funders were involved in writing Proximal Origins. They commented on how they wanted the wording to change, which parts should be eliminated. They were not mentioned either as contributing authors or in the acknowledgements, which is absolutely essential to assess basically whose perspective you're reading in a scientific article. So there were a lot of things that really shine a very, very bad light on the entire field and these earliest
Indeed.
applications.
So I interrupted you with point two. So carry on with what you were going through before we did that little diversion through circumstantial evidence. But I think it's very important for people to understand that there's different grades of evidence and there's different categories of evidence, and there's now
Absolutely.
also time and evidence. So once you start painting a picture with multi pixels and they coalesce, you've got a burden of evidence rather than a burden of doubt. And I think the probably the final nails in the coffins, and probably you're about to talk about this, is the specificity, scientific specificity of those nails. Which, so now we basically said, wow, there's a huge amount of circumstantial evidence that is very hard to ignore, and yet it has been sort of either ignored or sort of deflated or boxed into, oh, conspiracy theory, people doing research on their own, Googling stuff, they don't know anything. So now comes probably the much more sort of significant and weighty evidence, which is the genetic of it, the molecular, aspect of that evidence.
Yeah, exactly. So let's start with that second point that Rina Chan made, the furin cleavage site. Sorry for introducing another probably difficult to understand word. So let me try to explain it as easy as possible. So furin is a so-called protease. It's an enzyme, a protein, the building block of life. Yeah. Most of our cells are made out of proteins. They're encoded by DNA and translated in mRNA, which then produces specific proteins. And this one can cleave other proteins. So it's like a molecular scissor, so to say. And it plays a very big role in many processes, furin. It activates, for example, hormones, or it activates receptors and a lot of different pathways. It actually plays also cancer to some extent, makes it worse. But in virology, it's very important to have the so-called spike protein cleaved so that the virus can enter a cell. So the role of this protease is basically when the spike protein has two parts, S1, S2, and the S1 basically sticks to the receptor on the surface of the host cell. In this case, it's predominantly human ACE2. And then this doesn't really help the virus to get inside of the cell because just being stuck out there with two basically vesicles, one is the cell membrane and one is the viral membrane, which don't like to get into touch easily, doesn't help the virus if it can't put his genetic material inside of the cell. So if you then cleave some of the spike proteins, another part of that gets accessible and gets activated, which basically punches into the cell membrane and pulls the membrane of the virus very close to the membrane of the host cell. And this then allows the virus to inject its genetic material into the host cell, where it can be translated, and where basically then all the viral proteins are produced, and in this case the RNA is replicated, and where again the membrane from the host cell will in the end up as the viral membrane for all the billions of daughter viruses. And there's other proteases that can do that. So one maybe some people have heard of is, for example, trypsin. It works in your intestines. It's basically helps to chop down all the proteins that you eat into small pieces so that you can take it up as nutrients. And a lot of these viruses actually viruses that live in the gut of bats. So trypsin can absolutely do the job there. And it also limits basically which sort of tissues this virus can infect. Because trypsin is not produced, for example, in neurons. A virus that relies on trypsin could never infect a neuron, could not cause very severe damage in you. But furin, for example, is expressed in a lot of very vital organs, for example, the brain, but also, for example, in the respiratory system, which allows these viruses to be activated if you inhale them, which of course is much more likely to start a pandemic nowadays, where we all wash hands and use toilets and sanitary systems. So it's much more difficult for a virus to spread from, for example, feces to the next person. Yeah, and because of that, scientists have realized this quite early on. There's other viruses that are not directly related, such as MERS. It's also coronavirus, but from the MERS-related family. And those have the serine cleavage site, and this makes these viruses very lethal. And then, for example, in bird flu, for example, it has been known that the most lethal strain have a furin cleavage sites, while the others don't have it. So virologists started playing with that. They put furin cleavage sites into, for example, bird flu viruses that don't have it yet. And then they saw if they put that into a harmless strain, then this harmless strain could start killing chickens and could infect their brains and hearts and so on, which was completely impossible before. So the furin cleavage site is really basically something that has been played with a lot in this field in synthetic virology. They put this also into the only case this was ever seen in a SARS-related virus, in a sarbecovirus family, towards which SARS-CoV-2 belongs, and there's about 1,500 viruses here. And before SARS-CoV-2, none of them had this furin cleavage site, except for one spike protein where they put it in a lab, basically. So the only other example is a lab-generated virus. And we also know that, for example, Xi Li and Ralph Barrett collaborated on a project where they added the furin cleavage site to a MERS-related virus.
And
Yeah.
just a little question, because I've heard this before when Ralph Berry gave an interview with a Italian scientist while the pandemic was going on. Very early on, I think it's 2020 or 2021. And she basically asks him whether he is the father of a procedures in which you can insert things without leaving a signature of insert. And he proudly confirms that, yes, in fact, he is. So my question to you is that not the beginning of plausible deniability on almost anything that has that directional probability in terms of lab versus natural zoonotic? If you essentially have, is it significant enough, in other words, what he was talking about in synthetic biology and, you know, gain of function, even if it's for development of so-called vaccines? Is it a significant statement to say that, oh, and by the way, there's also a way that I've invented, which is that you won't be able to see the signature of an intervention, a human intervention into that?
Yeah, yeah. So basically, this is, let me go a little bit back. This was a technology that was invented also for molecular cloning. They use this, for example, for making genetically modified plants. And it's basically this relies on a specific type of restriction enzymes, which are enzymes that cut DNA in a very specific manner. And those so-called type 2S enzymes, they cut it not directly in the recognition site, so that each end of the fragment that you cut has part of the recognition site. If you fuse them again together, you will still see the recognition site, but they cut directly next to it. So you can use these restriction enzymes to make whatever restriction site you want. And basically those have a short sticky end. So the DNA is two, basically two strings, right? And if you don't cut them straight, but you cut one string a few nucleotides upstream and the other one further downstream, then this produces a so-called sticky end. Yeah. For example, you could have a TTAA sequence here, and if you have another end which has an TTAA, those will basically pull each together like a magnet because they have matching nucleotides. And this helps basically to put large genetic structures into the right sequence. For example, they have religated or recluded together 50 parts in exactly the right sequence in a single step with this method. So it's very efficient. It's very easy to
Right.
design. You can put these restriction sites because they're not in the final product. It's called no-seam in virology. You can
Yes.
put
Yes.
them wherever you want. It's not very challenging to design those, but there are downsides. But I think people in the field were aware of this technology. It was used quite a lot. And that's why they thought no one could prove this comes from a lab. But actually, for the very specific project they planned in the fuse, it has major downsides to use this technology. And the virologists that gave these early statements saying that it's certainly from nature, in private, they discussed and said, we will never be able to prove this unless there's someone from the Wuhan lab who comes out and basically as a whistleblower says, oh, we did this. There's no way to prove this. But it was not
Right.
done with this virus, is what I believe, or what we have later published in a preprint and
Right.
tried to get into a scientific journal ever since. So there is this technology to make this completely invisible, almost completely invisible, depending a bit on the viral strain. Yeah, and if you look at that SARS-CoV-2, the furin cleavage site is not only unique, it's not only the only virus that has that, or 1500 or 800. It looks inserted. So basically the parts right next to it are very similar to related viruses, and you just have these 12 letters inserted that code for exactly these 4 amino acids, a lot of R's that are needed to make this furin cleavage site. And we can, for example, cite Robert Gary from his internal emails. He said then in private, it's a perfect insertion that adds the furin site, and that he can't think of a plausible natural scenario where you get from the bad virus or one very similar to SARS-CoV-2, where you insert exactly these 4 amino acids and 12 nucleotides that have all to be added at exactly the same time to gain this function, and that without any changes in other amino acids and S2. So basically, he says it looks, of course, evolution could lead to something like that, but evolution would be not as clean, as precise, and would also lead to other mutations, for example, in the second half of the spike protein at the S2. So in private, he says this is clearly engineered. Then he published the direct opposite as the main responsible author in one of the leading journals. So I can provide you with a alignment if you want to show resources. Also, I made a talk about this. You can just put my name into YouTube. I have, I think, 80 references or something in there. Almost
Right.
everything I say, except for the most recent data, is there. And I
Yep.
plan to publish another Substack pretty soon where you find basically all the other references. So yeah, it could have evolved in nature, the furin cleavage site, but there's also another point there, that in the diffuse proposal, they say not to add any furin cleavage site, but a human identical one or human specific one. which is not really a thing because this is a very ancient mechanism. So furin from a bat is the same sequence as furin from a human or from a mouse. But they probably envisioned a scenario where a bat virus without that infects a human. And then there's a recombination event where human mRNA gets integrated into the bat genome, and you get a human-derived furin cleavage site at exactly that section. And then this could basically make this virus much more infectious and lethal. And the suspicious part here is, or one of them is, that this furin cleavage site matches an 8 out of 8 amino acids with a human-specific one, which was also published, I think, 2 years earlier at UNC, where Ralph Baric was working. So it's very likely that he was somehow in contact with his colleagues from almost the same department next door, that were also looking into these very specific proteases. So it made a lot of sense. And then a bit later, after the fuse proposal, mostly through efforts of US Right to Know, we actually got access to the drafts of that proposal. And in those drafts, we had a figure showing this furin cleavage site at exactly the S1, S2 interface border in the spike protein. So basically, it's like a diary where you have a guy saying that whatever, if he sees his ex-wife again, he will kill her and bury her under the tree where they first met, something like that. And then you find his ex-wife under that tree, and it's just very difficult to explain that if she died from a natural death. Yeah,
Indeed.
something in that way. Yeah, you basically finding the diary that lays out exactly the type of modification that we find in this virus. Yes, I think. Yeah, this is we're still at the very beginning. Maybe we have to speed up a little bit. The third key point was biosafety. But if you have any questions regarding the fume cleavage side, we can also.
No, no, no. I just wanted to have this as a way to check that I had understood correctly that this is
Absolutely,
yet another
absolutely.
problem.
Yeah. Yeah. So the third point that Alina Chan made was biosafety. I think it's very, very important to notice people always discuss this biosafety level 4 lab in Wuhan because it's the only biosafety level 4 lab there. And they were saying it's far away from the market and it's so safe. They have all these pressure suits, several layers of gloves. The whole building is basically airtight and within another airtight building. So it's very unlikely that an accident happens there. But she gave an interview with Science, and in that she said that they handle these bad coronaviruses, which are much less lethal than, for example, MERS and Nipah and all the others, at biosafety level 2 and 3. And that is a huge, huge difference. Biosafety level 2 is basically like a dental office, kind of, yeah. So people wear lab coats, yeah. They wear gloves, they wear glasses, but they don't even have to wear simple face masks. So what you had to wear to go to a supermarket to buy food, they said, "Oh, we don't need that to work on these viruses." And we know also from the diffuse proposal that the US colleagues were aware of that. Peter Daszak was very proud that they could generate these viruses at biosafety level 2. It would save them so much money. And then Ralph Baric said, on a side note, if you tell that to the US people reviewing this grant, they will freak out because it's insanely risky. And then basically they crossed out the 2 and put a 3 there. And later there was another side note where Peter Daszak said, well, once we have the grant, we basically will do a lot of work in China. But for now, we say we do it in the US. Because that will make the reviewers happy. Once we have it, we can still, argue with the granting organization that we have to do something there. And don't worry, Xi Li, we can do a lot of this in your lab, basically. And there was no objections. Yeah, Ralph Baric never said it's insane to do this. This could kill millions of people. He just said, no, we should not communicate it like that to the reviewers of that grant proposal.
But also, I mean, the one thing that I think screams at is, even if you're not a scientist, even if you do not understand that between BL2 and 3 and 4 there are huge differences, even if you leave that aside, the concept that you would, as a country that has really complicated, and I'm being very diplomatic, relationship with another country, another large country like China, that you would plausibly entertain the idea to do high level biosafety research with no oversight, because it is a particular structure government wise that doesn't allow you to do that. And it is a military city, and the military oversight of that lab was also spoken about. And that you would consider that even if everything goes well and they did it in the right lab, and everybody did exactly what was in that grant, this the simple concept of entertaining that idea, isn't that just geostrategic madness? That you would say, and at no point will these people sit back and go, you know what, now we get it, we know how to do it, we'll do our own thing, you won't have any supervision over this, and since we have the sample anyway, and now we know that you told very tall tale to your own supervision, we can put you into a world of pain. I mean, a 10-year-old would know this if you were in the schoolyard and fighting your enemy at the time. How is it that so far high this hierarchy of alleged experts and incredibly intelligent, one has to assume, people, that this is not the first shutdown that occurred, which is nope, this doesn't happen there. Doesn't matter what the cost is, doesn't matter what the savings are, you just don't do that. I mean, is
Yeah.
this just me or
No, I basically ascribe it mostly to human arrogance, right? And then you have so many players involved, and they all sometimes you have these things that then everybody trusts that someone else will, warn them if something bad is going to happen. But scientists, even if they're like absolute experts, they're not necessarily very good at judging the risks involved with their work. For example, there were these demon core experiments after Second World War. It was the I think plutonium core that was identical to one of the atomic bombs they used, and they didn't need a third one, so they gave it to scientists. And basically, whenever you put some reflective material on the core, it can go supercritical, basically explode, because the radiation reflects back into the core and can start a chain reaction. So if you just leave it open, it's no problem. But then one scientist there had these reflective spheres, and he couldn't find the spaces to leave some gap in between so that some radiation could out. He put a screwdriver in there. It fell out. And it got super critical, and he could somehow tip it off, but he got lethally irradiated with that. He killed himself, basically, although he completely knew about this risk. Other Feynman warned them that this would happen just before. And then I think a few weeks later, another scientist put these blocks around that are also reflective, I think beryllium blocks. And he drops one because he doesn't want to use the safety system from his hand and completely shuts off the core. It goes supercritical again, kills him. The guard next gets leukemia, dies a little bit later. So these are absolute specialists. Yeah, they are the world elite in nuclear science, and they within weeks kill themselves because they misjudge their basically their capabilities or how likely errors can be. They don't use safety precautions that are in place because it would have taken a few minutes longer to prepare them, and they risk blowing up the entire city, basically, right, with a nuke after a year after they saw what kind of destruction nukes do. Yeah. So this is just I think quite telling that scientists, although they are smart and know a lot about this stuff, they sometimes, if you work with something for your entire life, you start misjudging the risks involved. And here we're talking about a virus that, without these modifications, would be completely harmless. Right?
mm-hmm.
Nucleoside couldn't cause anything other than maybe a day of diarrhea. In my opinion, and it can't kill any hamsters if you remove that site. It's completely harmless, and basically, it's it has happened in other fields before. Yeah? You start with a harmless virus, then you add one more risk feature, and then you add another one, and it's very very difficult. Or they were maybe just too arrogant and thought that they could predict this. And from the US side, I think they wanted to have. I'm speculating here. I think they wanted to keep an eye on what the
Hmm.
Chinese were doing in their biosafety 4 lab. So they wanted some US reports being sent to the US and some people being in this lab that could look what the other scientists are doing there. Maybe that was the intention, to monitor their potential bioweapon activities. This is very difficult to judge, but
Sure.
it's absolutely outrageous that they worked on these viruses at the safety level where not even masks are required. And even if you look at the higher safety levels, 3 and 4, there was a study from the US where they had 800 cases between 2006 and 2013, where workers had to get medical treatment or evaluation after an accident at these biosafety level 3 and 4 labs, where you really have this high pressure suits and these enclosed labs. So basically, one accident a week or something like that. Even those are not that safe. And the other thing that's very different about SARS-CoV-2 is that it doesn't always cause severe symptoms. People could infect themselves and just, go about their lives and maybe infect someone else. People are under pressure. They may have to finalize their thesis or have to get some data for the grant where the deadline is very soon. So if they maybe have a cold, they still come to the lab or don't isolate themselves, or um, errors happen. I mean, humans are humans, right? I think uh, the major fault is not even here on the PhD student who does this, because he can't oversee all the consequences and cannot trust that the PIs and the regulators say, okay, this is ethically approved, that's safe to do, or like worth the risk or whatever. Yeah, but we just have to make sure that people are no longer under the impression that this work was done at high biosafety levels. It absolutely wasn't. And there was a biosafety level 2 lab actually, I think 300 meters from that market, where they found many of these early samples. So uh, it could have also come from any of the biosafety level 2 labs, of which there are many in the city. Yeah, shocking, really shocking to see how careless they were and how little precautions they put on themselves to save some money. Right? I mean, we have receipts from Peter Daszak, where he needed 20,000 euros to fly for a conference, first
Yep.
class. Yeah, that
Yep.
could pay Chinese scientists for a long time to work at biosafety level 3 and to prevent a pandemic, but yeah, this was not the priority there. Yeah, it had to be cheap and had to be fast, and like a lot of things get then outsourced to countries where safety is, which is of course a huge cost driver. If you have to put yourself into these one-time use suits in some cases, yeah, every day, takes a lot of time, takes a lot of months of training to really do this safely. There's also actually a note from Wuhan that they were lacking trained experts to work in these labs, and that didn't have enough of the biosafety equipment. So but yeah, as they didn't work on these fairly harmless bad viruses before you manipulate them anyway. Yeah. It's not unique in this field. You also see basically the regulations for sampling these bats which harbor these lethal viruses are very strict. You have to wear personal protective equipment, goggles, gloves. And then you see also Western scientists, virologists, professors that on their social media post pictures where they basically have bats hanging from their bare hands,
Yep.
and where
Yep.
they, from their classes, uh, and where they go barefoot into these caves that are full of bat guano. And you really kind of, I don't know, they put them online voluntarily, and it just doesn't make sense. Either these are highly risky viruses, then you don't go into these caves barefoot, and or basically their work doesn't make sense because naturally those viruses don't jump or not so dangerous, and then whatever. Yeah. But it's insane. Whenever you don't have good oversight, people tend to get very lax apparently on safety. Yeah. It's not a happy topic. It's really depressing. No, but
it's important that you get the points that both she make and a lot of people make, which are basic point, and then that you add your scientific opinion and expertise onto that as well, so that for the record it stays there. And then later on, depending on how the future goes off with the gain of function regulation, this is essentially part of that edifice of knowledge, right?
Yes, absolutely. I think this is my role. I'm kind of like, the molecular engineer. I know a lot about the technologies and understand the principles. I mean, what we talked about so far is just, everybody can understand that wearing face mask helps. If you handle tons of viruses and manipulate them in a lab. But this is my key incentive to provide this kind of like forensic expert opinion on this very specific DNA manipulation and evidence that we see within the genome, and to leave it up to people to form their own opinions, but after they have listened to the facts. And I really invite everybody to check everything I say. And please point out if I do anything wrong, if anything I say is wrong. Of course, I'm happy to correct myself. Yeah, the next key issue would be the papers that pointed this towards the Huanan Sifu market. I think there's a lot of in the article in the New York Times. People can read that for themselves. It's also so in one of the articles that basically claimed that there must have been two jumps, so two separate events where this virus got from an animal to a human, based on these two early lineages. This article contained major coding errors, which was later found out, although it was published in Science. And even if you ignore that, if you just focus on their key assumption, which is that these two lineages differ only by two mutations. So there's no way basically to saying this is a mutation that occurred in an animal and this is one that occurred in a human. Yeah. Basically, if you say that whenever you have two mutations in between two viruses, this comes from two different jumps, then we would have seen 11 jumps at this Diamond Princess cruise. This where basically there was this SARS-CoV-2 outbreak fairly early on, and where a lot of the viral sequences were sequenced afterwards, and where we could directly see how related they were. And there are a lot of people that had two mutations in one patient, and sometimes even three. And so saying that two mutations are indicative of two species jumps just doesn't make a lot of sense. And a lot of virologists also said that that doesn't make sense. Uh, and even even if you ignore that and look at the proximity, which was the other paper saying that all these samples were in direct proximity of the market, and it's very, very unlikely that this happens if this biosafety lab, which is far away, was the source. If you show biosafety 2 labs in the map, they're basically spot on in the center of these clouds they generate with the samples. But there's another critique here by Professor Michael Weisman. He argued that people connected to the market would basically got infected there and infect other people, which in average don't live as close to the market, or they should, in some cases, go to work somewhere else or come there. And he analyzed how close people that had direct contact to the market were to the market versus those that didn't. And the opposite was seen here. Those people without direct contact to the market were actually closer to the market, which is just doesn't make much sense for an outbreak when it starts at one location and then it spreads out. I think this is
Sure.
also fairly obvious. And then I think also from a logical perspective, everybody who's been to a major city, it's I think more than 10 million inhabitants here, to say that after a month or so to pinpoint a specific location, to 3-4 kilometers further south or north, in a city where people travel probably 15 kilometers on average every day to get to their work because the houses are cheaper on the outside, yeah, where they use public transportation, where millions of people are in contact in in the trains, in the public transportation, and in the supermarkets and so on. It's just impossible to track that down to a few kilometers. And if you do that, why do you ignore that the viruses are naturally found 1,500 kilometers further southeast, and ignore that they miraculously got to Wuhan in the first place, and then focus on these 3-4 kilometers and basically a black box of 3 months, and then all the data you get is provided by the Chinese Communist Party, of course. If they
Hmm.
want to tell you that this is close to the market, they only show you data close to the market. And we have actually some documents also where, because of SARS-1, again, they knew it could jump from civils, so they thought of course, where do we have animals? Where we find animals, of course, there's this big market. And that's why they sampled a lot there, why they collected a lot of samples there, and why when they had limited resources for sequencing, and they said, oh, well, let's look at the patients first that have any contact with that market, right? So this basically explains this spotlight effect, where people basically look for the keys under the lantern because this is where there's light and they can't find it where it's dark. But this doesn't mean that that's where the keys is. It's just a bias, a sampling bias. Basically, you have an idea that could have jumped from an animal, so
Yeah.
you go to where the animals are. Yeah, so very weak
So in
evidence.
addition, yeah,
Sorry.
in addition to what Elena found, then what are the things that for you are very important to add on as we're concluding the episode to make people really understand the depth of this evidence sort of coalescence, because I think that is something that probably isn't covered in the general press or
Yeah.
far less the corporate media.
Yeah. So I think her fifth point, that there's absolutely no evidence for a zoonotic event, is very strong as well. We found those for SARS and MERS, and there's not a single animal up to this date, which is very unusual because the technology got so much better and so much more sensitive. I think that's quite strong. Um, I think what she didn't mention are actually points that are not as well debated, not as much debated, but are also actually stronger. So, for example, all these virus samples we had at the market were not ancestral. So, if you look at the mutations, they were derived, they were older samples. But we had one of the two possible ancestors for all human lineages, and this was actually sequenced um, and accidentally released, so to say, with sequencing data from a completely unrelated experiment. This is a preprint by Sabai et al. Um, and the virologists tried to basically, they didn't allow to upload this preprint to the most important server, and they tried to delete the respective samples. And in these samples, you find that these mutations that make this virus most ancestral. Also, some later mutations, and you find DNA of a cell line that is mentioned in the diffuse proposal. So, if we assume that this was a lab accident, then the most ancestral genome is found in presence of the so-called Vero cell lines. And Vero cell lines, as opposed to all the raccoon dogs, we know raccoon dogs are very hard to infect with this virus, even with a fitter later variant where they tried this. Basically, the infection died off. The second generation animals that got infected had tiny amounts, like 600 RNA copies per milliliter, which is nothing. This is like 10 million fold lower than what you have in humans. So it's clearly not a typical intermediate host. You check all these boxes with viruses. They're in Wuhan. They are highly susceptible to these viruses. They're very easy they are basically mentioned that they wanted to use those in the proposal. They are in close contact with scientists, of course, who handle them, and uh, basically Even the sequencing facility where this data was generated, is called Sangon Biotech, is mentioned in the FUSE proposal. Yeah. So again, too much of a coincidence, in my opinion. Yeah, I think what happened here is that they panicked. They basically saw this virus circulating in the city. They took all their samples from related experiments, sent it to their sequencing facility, which they used for years. And then they got all their results back. And then they saw, okay, some passaging experiments led to later mutations, but some other experiments really had the ancestor virus, which they produced in these virus cell lines. And then they, of course, didn't publish any of that. But other people, which used the same machine, published their results. And through contaminating reads, people could piece together the entire genomes and all the genetic material from the cell lines. And basically, in my opinion, this is much, much stronger evidence for an intermediate host because it's ancestral. It's the earliest known version of this virus that is in circulation. And again, it's exactly at the company they mention in their proposal. Another point I think I find very, very interesting is that this virus looks like an additional chimera for a part of the spike called receptor binding domain. So basically, this is the section of the spike protein that sticks to the surface of the cells via ACE2. It directly interacts with the human receptor ACE2. And if you compare SARS-CoV-2 with, for example, Red 13, the other virus they had in Wuhan, and you check which parts of the two viruses are very different on protein level, the first thing that sticks out is the furin cleavage site. And depending on how big you define, it's like 50-80% difference. The rest of the virus is like 2% different. And then the receptor binding domain, again, is more than 20% different. And they mentioned in the diffuse proposal that they wanted to make these chimeras for the spike protein, for the furin cleavage site, and for the receptor binding domain. And the Reg13 virus doesn't have a receptor binding domain that could infect humans. Yeah, it's very inefficient. And they say in the proposal that they want to screen other viruses and predict via 3D models which of the receptor binding domains fit best to the human receptor, and then singly and in combination put these receptor binding domains and or furin cleavage sites into coronaviruses that they collected in bats. And this would completely explain why this virus spread like wildfire. It's completely new to this type of viruses. If they jump from a natural event, they usually need years of adaptation to humans before their proteins are so well adapted that they're very efficient at infecting one human from one human to another. Completely different. In this case, this virus was basically completely unstoppable. And if they did that, if they modeled the receptor binding domain and predicted those which bind best, and then put those in, and Reg13 like virus, this is actually the closest you can get in a sequence. If you take the Reg13 sequence, you add a furin cleavage site, and you add the receptor binding domain from, for example, Banal 2052, this is the closest virus that you have in nature. So it's basically a double chimera or double mutated virus with the furin cleavage site as well as the receptor binding domain. Again, I mean, what are the odds, right? And then another thing that was really striking to me is that they leaked plasmids coding for the SARS-CoV-2 spike protein in 2019. There was later basically a guy from Spain programming an AI tool to screen the huge databases for all the DNA for SARS-CoV-2 spike-like sequences. And they found that there was a plasmid, so a DNA segment that you use to produce tons of protein, for example, in which they had put in exactly the same sequence of a spike protein as you see in SARS-CoV-2 without the furin cleavage site. So the furin cleavage site was not in there yet. And this was found in 4 patient samples collected in 2019.
Wow.
Some virologists argued that it could be a later contamination. They collected a sample in 2019, it was sequenced later. the plasmids got in the lab when the samples were prepared for sequencing, but the samples actually, the proteins contain a lot of mutations, and those mutations don't occur in bacteria you use in labs, because you remove all the genes that destroy your DNA, and whenever you have a mutation, you throw away the DNA.
Yeah.
And so it's basically impossible that these DNA pieces that coded for exactly the SARS-CoV-2 spike protein were coming from a lab. They must have been in a, like, they were produced in a lab, but they must have been in a bacteria in the wild for some time. And if you then look again at the diffuse proposal, there's a few other modifications. So they add, for example, a trimerization domain, which helps to form a trimer of the spike protein, so 3 chains connected. And in the diffuse proposal, they mentioned that they want to use exactly this technology to generate a vaccine for bats. Also, the plasmid looks like something you would use for a bat vaccine. And the leader peptides and everything, it's clearly synthetic. It fits 1-to-1 to what they wanted to do. And it was sampled in 2019, where officially no one has ever heard of the sequence of the SARS-CoV-2 spike protein. Yeah. And then the last and probably key point is, or like one of the strongest ones, what we tried to publish in a preprint. I did that together with Alex Washburn and Tony Van Dongen, both in the US now. We were looking at how the Wuhan Institute of Virology had generated their synthetic viruses before, which methods they used. And in 2017 paper, where they also wanted to generate a lot of varieties of one virus, of their synthetic with one strain, and they wanted to put a lot of spike proteins in there. They used these 2 enzymes, BSA1 and BSMB1. So we focused on those and checked if there's anything suspicious with regards to those enzymes in the genome of SARS-CoV-2. Also looked at a few other enzymes that are really good to do these viral assemblies, but those didn't light up. And if you make a virus in a lab, usually, or what they did is that they use DNA segments that are not too large, so you can just order them up to a certain limit. In the major provider, this is 8,000 base pairs, 8,000 letters. You can just go there and tell them, I'd like to have this DNA sequence. It's very, very cheap. So making such a virus is probably less than $5,000 these days, which also shows how insanely risky this technology is. And they said, okay, please give me these 6-7 segments. And then if you put these sticky sites, these restriction sites at the right places, you can assemble those segments in a lab. And then have a new virus. And we basically looked at where those stitching sites, how they are distributed over the genome. And we found that this looks highly suspicious in SARS-CoV-2. None of the other viruses had a similar pattern where you now have one restriction site always in almost equal spacing. And then they also used these 2 restriction sites, so that they had a specific one flanking the furin cleavage site and receptor binding domain, so that after they had completed their first virus, they could just sequence that and verify that everything is correct on DNA level, and then go back in and make a variant without the furin cleavage site, with a different receptor binding domain, with the furin cleavage site from another human protein, and so on. They mentioned that they wanted to put like 7 to 8 different receptor binding domains and some number of furin cleavage sites into these viruses. They wanted to make 3 to 5 new viruses. And this is the point where using this no-seam technology gets very inefficient, because then you have to assemble the entire virus from scratch, from the beginning, for every variant you want. And those ligations can go wrong. Sequencing is expensive, and it's much more efficient to just assemble everything once and then leave those restriction sites in, and then just surgically cut out that one part that you want to modify and put in a new variant. And that variant would then cost only $200-300. Yeah, really, really cheap. Um, and this is basically, I think, what the virologists didn't realize in the beginning, that in the diffuse proposal, they were not looking at making one or two viruses, but 15, 20, 30 variants of new backbones and 3 to 5 new backbones per year. So the scale of viral production was just unheard of what we have seen in other projects before. And if you want to do that, you don't use no seam, and that leaves then traces. And this is what we found. And the key evidence that we looked at was not even the spacing. I mean, the spacing can occur naturally. There are like 1% of SARS-CoV-like viruses have similar spacing, where you have just a similar pattern that you could assemble in the lab by chance. But if you want to introduce these stitching sites, you always use silent mutations, so mutations that only affect the DNA and the RNA and not the protein sequence, because that could make a virus dysfunctional. We have seen single mutations, D614G, which completely affect the efficacy of a virus. And if you do that in a lab, all your colleagues would say, well, you put in this mutation, you have a different protein, maybe your virus is no longer dangerous because you changed the protein sequence. So they always use silent mutations that don't change the protein sequence. And that makes it more difficult to put these stitching sites into the genome, which is why the pattern is not perfect, not as perfect as with no seam. This is technically just impossible. But when we look at how many mutations you find in these stitching sites versus the rest of the genome, then the number is massively increased. And these sites are only used to assemble DNA, and we're talking about an RNA virus. These enzymes are not present anywhere in humans or in nature. In nature, they come up in some bacteria, so they don't play any role. So there's no explanation why a natural virus would specifically mutate these DNA restriction sites. And the chance that you have this many mutations exactly in those sites and then far, far less mutations everywhere else is like less than 1 in 10 million. This is the key result of our preprint, basically showing that this was more than 10 million times more likely a product of targeted manipulation than random evolution. And actually, this is the part that gets ignored always. We had like hundreds of virologists insulting us, commenting. My own institution wrote a comment press release, for German media, in which they said, well, the methods look solid, but they made a lot of mistakes. And they don't even mention the key finding. They just completely ignore that. I asked them
Yeah,
afterwards.
because yeah, that's the elephant in the room. That's the thing that is probably out of after listening to you. This last bit is probably the bit that is jaw dropping. You know, evidence that how else would you explain that? And the malfeasance that goes with it is sort of jaw dropping because you could feel like it's almost a machine gun out of control where it the engine of both hubris, we've done it before, nothing was really happening, this concept of it's cheaper, at the scale of what they were doing, what had defined cheap, right? So you playing literally with incredible nuclear power,
Yeah,
essentially, and the
exactly.
odds of getting this wrong are just accumulative with every sort of roundabout things that you do, including in the genetic part of it, in the engineering part of, oh well, we need more, we need more support to be able to do this even better and faster and cheaper. That in itself is probably the definition of unregulated madness and unstopped madness up until the epitome of this, which is a massive pandemic that's killed millions of people, that's cost trillions of dollars, and that have changed probably the face of the earth for and the human genome. One has to assume because of both the virus itself, and then after that the vaccine, mRNA vaccine, that were based on somewhat of the same problem.
Yeah.
It's jaw dropping. I mean, literally, it's jaw dropping. So to conclude this, maybe we'll have to do another episode on gain of function itself, because we talked about this particular event, which we're still in the middle of, as most of the countries that have been heavily exposed to the virus and heavily vaccinated are seems to have recurring COVID right now, whether it's Australia, Canada, Europe. So people have been affected, continue to be affected. For those who are not dead, for those who do not have disease associated with this, and so to conclude this, I would like to ask you, what is it that you, if you had a absolute power, an absolute scientific say, what is it that you would want to see right here, right now, not in weeks, months, but right here, right now, as existential crisis decision um, for those for those people and those institutions that are capable of delivering this?
Yeah, first of all, I think we really have to have a serious investigation. Yeah, this is absolute number one. I'm a guy at my computer and far away from this. There's much more that can be learned from going into the labs, seizing their computers. We're talking about World War scale of human loss of human life. Yeah, I think World War One was like 17 million people that died. And I think we are doing exactly the same mistake. We're thinking it's a one-time event. It won't happen again. It's just inconvenient. Nobody wants to talk about it anymore. I completely get that. It was hard for everyone. But if you don't, the risk of not getting to the underlying causes and to the basic of the problem is that it will happen again. And if you look at those outbreaks that happened afterwards, for example, the Ebola outbreak in 2021, it's also an RNA virus, and it's a virus that did not accumulate any mutations for almost 5 years. It's just biologically impossible that any virus in nature that is an RNA virus stops accumulating mutations and stays infectious in any host. But this is exactly what they wrote in their Nature paper. Um, and it wasn't very consequential. I don't know how many, like just a few people died from that outbreak, but it is just, again, almost obviously clear that an RNA virus that doesn't mutate for 5 years is either frozen in a lab, or reproduced after an old sequence in a lab. And the number one patient here was a nurse. She worked with patient samples. She had access to the freezers. Those samples are frozen before they're sent to the lab. I mean, nothing of that is malicious. Yeah, it just looks like she probably thought a sample that was not properly labeled. Maybe they didn't know the patient had Ebola when they took the sample. Maybe they didn't burn it properly or something like that. But if we keep looking away, exactly the same thing will happen that what happened with for example, the Catholic Church with child abuse, right? We only listened to those that had conflicts of interest. And the consequence was that a lot of people suffered. A lot of lives got destroyed and severely impacted. And I think exactly the same will happen here. We should not at all underestimate how likely it is that something identical to this will happen again, because the technology is so new and so cheap. Yeah. Building a nuke, if you Google the protocols or like the materials you need, you probably have secret services at your doorstep. I don't know, but it costs millions of euros, and those killed like 20,000 people, I think, 40,000, something like that. This virus killed 28 million, according to some estimates. I don't know how many of those are really dying with or from the virus, but the damages are huge. It affected the entire world, and it costs probably less than 1% of what costs to make a nuke. Yeah, we cannot underestimate the danger of this very new technology, and we have to regulate it. We have to have something like the IAEA that supervises use of nukes and um, nuclear power in all the countries that use it, that goes into every lab, that checks all the producers of genetic sequences for if they produce something that's viral or it's gain of function. And we have to, until we really got to the bottom of this, or I think for forever, we should really ban making pathogens that could kill humans more pathogenic, because safety is an illusion. There will always be some sort of accidents at some point.
Yep.
And if an accident kills 28 million people, and we can't even foresee the long-term consequences. With HIV, I mean, the first people dying were 10 years after the first
Yep.
infections, right? We don't know what this will do in the long term, how often we will get infected, what it,
Yep.
yeah, what kind of increase in autoimmune diseases and everything else this could cause.
Prion disease, brain disease, yeah. Brain degenerations. There's loads of stuff.
Absolutely, loads of stuff. And if you look at, for example, some of the viral samples on SARS-CoV-2 they collected in Wuhan contained these building bricks of synthetic viruses that code for Nipah, yeah, which is a virus that kills 70% of the people it infects. Yeah. So this was our, however you see this, even if you say, well, this was this was a whatever politically made pandemic, and I don't agree with that, but even then, the virus was unstoppable. Yeah, it's complete
Yep.
human hubris to think you can control that, and it's very easy to produce that. And if there's no oversight and no regulation in every country in the world, then this will certainly happen again. And it doesn't matter if this starts in a lot of countries that start building these labs now, like India or South Africa. Um, we have to be honest, and a lot of experts have to step back and say, okay, I was wrong here. I personally was wrong about not paying enough attention to this. I was working on cancer research, later on autoimmune diseases, which affect 10% of the world population. I heard on the side that this virology was dangerous, and then they made really dangerous viruses more infectious to humans. And I'm really, really sorry I didn't pay more attention to that. I wouldn't have changed anything. People still don't listen to me after I got my PhD and everything. Academia just doesn't really work like that. But
No.
we really have to be honest, and also journalists, if you had interviewed these guys without mentioning their conflicts of interest, go back, research their conflicts of interest, correct your statements. I don't care what you write about. I mean, I got insulted in so many major newspapers by people that we now know that lied, like, for example, Christian Anderson. And of course, he was a professor, so I really like to quote him. I don't care. I want my kids to live their lives and to grow up and have children and be safe. Yeah.
Yes.
So really, please do some soul researching and admit mistakes and this.
Yeah. Well, maybe we need to review the fact that competency in science at this level also includes character and ethics, and that should be in the CV just as much as how many wonderful achievements you've done in physics, mathematics, genetics, or otherwise. Because I think without character and without a fundamental ethics as a human, sort of a pro-human ethics in the sense of not just your role, but and not just your country and but the planet and the responsibility that you hold being a scientist with the power that you have. I think that needs to be reviewed as well because we clearly have selected or allowed for self-selection of calibers of people that are absolutely atrocious. So to conclude this
Mhm.
episode, and again, as I said, if you're keen to do another one, I would be very keen to have another one just on gain of function and biosafety and all the options for that. But I
Mhm.
would like to conclude this episode the same way I do with most all my guests, which is to ask you in this difficult battle that we're in, whether it's a battle for knowledge, it's a battle for ethics, it's a battle for survival of one's career, whatever it is, it's a very significant race. It's a very hard race. What is your weird sauce for that battle and that life that we have coincidentally found ourselves in at the moment?
Yeah, it's a very good question. I think, yeah, I'm not really a religious type. I think, to me, human life and human health is really something that's kind of close to holy. Yeah. I, from personal history now, what it means to lose someone, and what it means. I've seen people suffer, so I try to my whole life basically focus on curing diseases and making lives better. And this is, and basically, I think this is what keeps me going. Yeah, to see that some people who have the same training as me, who use the same protocols, or at the same companies, use this to cause this much suffering, and then to obviously lie about it and break all ethic guidelines with regards to publishing, with regards to being honest, with regards to uh, unbiased assessments in of the evidence, was really kind of heartbreaking for me, kind of destroyed my illusion of having a somewhat functional academic world. Um, and of course, I'll keep fighting, but we have to realize that everybody's resources are limited, and that at this point, I'll focus more on developing these therapeutics. I think people have the chance to, provide them with the evidence. If this is a democracy, people decide now, well, just go on with our lives, focus on soccer and everything else, then this is their decision. It's really up to you to write to your representative to become active. I've tried that. We've teamed up with 18 journalists, 18 other professors and scientists and wrote to the EU Commission. They didn't even allow us to call them for 15 minutes. You can't do anything as a scientist. I tried to approach politicians I had personal connections with. They are not under pressure. Nobody is daring to basically risk their reputation for something that is considered was called a conspiracy theory. And as long as people don't realize the threat of this, and not just this pandemic, that there likely was another lethal accident since then, and that it could happen again anytime, we are all in great danger here. But predominantly, I'd like to appeal to my colleagues to really step up. And after every talk, they say, "Yeah, you're right. Yeah, it's correct. But this doesn't help. Yeah, you have to go to a journalist, tell them, hey, something happened here. We have to talk about this. We have to fix this. This could, destroy the fabric of our society, destroy basically our highly interconnected pathways that provide us with food, with medicines, with technology, and to really step over your first maybe false impression and ignore what the consequence could be to yourself, and just be honest here once, because almost exactly 100 years ago in Germany, basically, we did the same mistake. Yeah, millions of people had died after World War One. Nobody wanted to talk about it anymore. Nobody wanted to look at the underlying problems with racism, with poverty, with nationalism, with patriotism, and so on. And we did the same mistake again, and it was way worse. And this is our chance to avoid another major pandemic if we come together and are honest. And I really hope people find it in themselves to do that step, and because I think we all have these challenging times in our lives, and
Mhm.
this is yours, and this is your time to really show what we are made of, and that we are better than this.
Dr. Valentin Bruttel, thank you so much for your time. And personally, I would like to thank you also for your courage and your contribution to science and to knowledge. And in my own little way, I try to carry the voice of people like yourself. I don't have a big mic or a big speaker, but if I did, I would promote that much higher. So hopefully, every one of us, those who are listening, can also do their own bit. You don't have to be necessarily a scientist. You just have to be courageous, brave humans who just have reached their limits of tolerance for the intolerable. So, thank you so much for your time and effort. And hopefully, we'll have a longer chat on gain of function at some point if you have the time. Thank you again, Dr. Rutel.
Be very happy to do that. Thanks a lot for your interest and for picking up this topic and allowing this other perspective to be basically at least discussed, and to allowing a scientific process that is absolutely needed here to inform those people that listen to you about what is actually out there, and that this may not be what they read in a newspaper, unfortunately. Thanks a lot.
If this conversation stopped you in your track, share it with your network. You never know whose life you might change for the better. Thank you for listening. Stay curious about our next guest and stay curious about life.